Hi, my name is Greta Rockstad, and I'm a master's student in the turfgrass breeding and genetics lab. And the focus of my project is on using high-throughput phenotyping methods to study drought stress in St. Augustine grass. And that also means that I can sometimes play around with the drones. So this is just a quick video clip of some of our plots in Lake Wheeler, which is also where one of my sites is. So just a little bit of background. We know that irrigation is the majority of home water usage. There's a lot of homeowner interest in cutting down on irrigation requirements, and municipalities are also interested in this. Sometimes their irrigation schedules, particularly during drought, you can actually be penalized if you irrigate on the wrong day. And furthermore, some places actually encourage residents to take out their turf grass and replace it with more native adapted species that use less water. But our lab recognizes that these native species can't provide all the benefits and functions that turfgrass can. So one big goal of our program is to develop drought-resistant turf grasses. The problem with this is a lot of turf traits are measured on subjective scales, like a visual rating of 1 to 9 for something like leaf firing or leaf wilting, and this makes pooling data between raters across locations and among different years kind of hard. So recently, more objective measures like digital image analysis and even more recently, drone imaging have been the way the field has shifted. So my project is focusing on how we can use drones to look at how well our ground-based measurements correlate with drone measurements. There have been similar studies done in Bermuda grass and zoysia grass, and that's what this figure below is showing. Along the x-axis, you can see NDVI taken aerially, and along the y-axis, you can see NDVI taken on the ground, and there's a very strong correlation, and we're hoping to see the same thing in St. Augustine grass. A secondary objective is to use this data we've collected on drought stress traits to map QTL for drought tolerance, and do this over a broader temporal scale than we've done in the past. To do this, we're evaluating drought in the field at 2 locations, Sand Hills and Lake Wheeler. The traits we're collecting include NDVI, percent green cover, and canopy temperature. These will be collected both on the ground and in the air. We're planning to do a baseline collection before any drought symptoms are apparent, and then every 3 days once the water is cut during the drought period, and then again every 3 days during the green up. To do this, the system we're using consists of a DJI matrice with 2 gimbals, the first of which contains an RGB camera, and the second of which contains a MicaSense Ultim multispectral camera. And this has bands so we can measure NDVI as well as canopy temperature with a thermal band. The third gimbal actually contains a downwelling light sensor that accounts for any changes in the lighting from one day to the next when we collect data. So if it's super sunny one day and then very cloudy the next, this will help to calibrate our readings. And on the ground, we have recognizable ground control points that we use to rectify the images in our processing pipeline. And finally, we have reflectance panels with known reflectance values, also to help calibrate our multispectral images. Because we're virtual, I thought I'd show you a little bit of the processing workflow that we deal with. So when we fly a plot, or if we fly several plots, we can generate anywhere from 200 to almost 1,000 images. And we start with the raw images and end up with something called an orthomosaic once they're all stitched together. And somewhere along the way, we generate a point cloud, which is what the first photo is showing. The blue squares are actually where the drone was when it took the photo, so you can see the elevation as well as the orientation. And then those are stitched together to form the next photo, which is an orthomosaic. You can see the blue flags on there with some words, and those are our ground control points. We put them in the corners of the plot for optimal rectifying. And then from there, it's easier to define the plot boundaries. We say, okay, the plot is 4 feet by 4 feet with 1.5 foot alleys. And then instead of wiggling around the plots, they fit really well in the picture. From there, we can extract the data on a per plot basis. And so this example is just showing percent green cover. And finally, once we have those numbers, we can use those to analyze the correlation between what we collected in the air and then what we see on the ground. And we'll do this within locations and between locations, as well as across 2 years. In the past, we've done some drought studies, but these have normally been, once we see visible symptoms, we'll start collecting data. But the hope with this study is to get a more nuanced picture of when plants start experiencing drought stress. So even though visual symptoms might not have happened yet, if we can analyze plant available water, we'll know maybe we can start taking canopy temperature readings earlier on and capture more of that temporal resolution. So to do this, we've installed a soil moisture sensor and a water potential sensor, both hooked up to a cloud data logger. And an example of this output is shown on the bottom of the screen in the graph. Along the x-axis is time, and there are a couple of y-axes showing things like water content, temperature, and matrix potential. So I'll draw your attention to the black box. To the left of it, you can see that matrix potential and water content was slowly decreasing, and the soil was drying out. And then somewhere between July 23rd and July 24th, there was a rain event, and everything spiked back up. So even though we couldn't see visual symptoms of the turf grass experiencing drought, because the matrix potential and the water content was decreasing, that might be a time where we should take canopy temperature to see if we can get any differences between our lines. And this is important because in the past, we've generally taken data at 1, maybe 2 time points when there's the most differentiation, typically towards the end of drought. But we know that there are several different genes that contribute to drought tolerance from the beginning to the end of drought period. So if we can capture data at the beginning, even before we can identify symptoms with our eyes, as well as in the middle, and then even when they're greening up, we'll have more power to say, okay, this region of the genome contributes to drought tolerance. Let's find a marker, and then we can screen our germplasm in the greenhouse, even before we take the time to put it in the field, and probably evaluate it for 2 years. And this is all part of our marker-assisted selection program that we're trying to implement in turfgrass breeding. And additionally, we've done some QTL mapping before in drought tolerance, and the results from this study will help to validate those results. So that concludes my presentation. Thank you so much for listening, and we hope to see you back in person next year.